Hello everyone. This is meant for class 11th biology students, and this video is meant for 6th July 2020. If we recollect from our memory, we know that we have been talking of anatomy of flowering plants, and in our previous sessions we have covered anatomy of root, both dicot and monocot, anatomy of stem, both dicot and monocot. We have compared both monocot root and dicot root, and similarly anatomically we have compared the monocot stem with the dicot stem. And in this session we are going to study about anatomy of leaf. And just like how dicot root, monocot root is there, dicot stem, monocot stem is there, similarly we have got dicot leaf, monocot leaf. So let's see the anatomy of that. And since we have already studied in morphology in our previous chapter, as you are seeing in this photo, monocots are having parallel venation and the dicots are having reticulate venation. This is how you differentiate monocot leaf with dicot leaf morphologically. But what about anatomically? Anatomically, when we take a section of the leaf, all of you will be taking it once you come to biology lab after your lockdown is over. So typical dorso-ventral leaf. A dicot leaf is a typical dorso-ventral leaf, and it consists of three main tissues: epidermal tissue, mesophyll tissue, and vascular tissue. Now when we take the section, this is how it looks like under the microscope. On top as well as bottom, you can see a pink layer of a single celled layer, and that is epidermal tissue. Upper epidermal tissue and lower epidermal tissue, that is adaxial epidermal tissue and abaxial epidermal tissue. And again, if you observe, over this epidermis there is a cuticle, both upper as well as lower. And as already said, the cuticle layer is meant to control rate of transpiration. Water loss can be controlled. So in a dicot leaf, we see one layer of epidermis, upper epidermis on top, and one layer of lower epidermis at the bottom. And we see, you can observe there, in lower epidermis there is stomata. Usually it is present in lower epidermis in dicot. Rarely you see in this upper epidermis. And as you all know, stomata has got guard cells, and the guard cells will be opening and closing the stomata, and this helps in two purposes: one, exchange of gases, and second, the rate of transpiration also can be controlled through this. And you have got second tissue, mesophyll tissue, and in this mesophyll tissue it is divided into two types of tissues. One, pillar shaped cells are there, you can see on top, palisade tissue, palisade mesophyll, and down sponge, amoeboid cells you can see loosely arranged. These are called spongy mesophyll. So mesophyll tissue is divided into two: a pillar shaped cells, palisade tissue, which will give strength and support to the whole leaf, and it has got a lot of chlorophyll, and having chlorophyll, it will make the leaf to undergo photosynthesis. That's the reason why it's green in color. And spongy mesophyll also will be having chlorophyll, but then look at the shape of the cells. These are amoeboid in shape, and you can also notice there are a lot of gaps in that spongy cells. A lot of air spaces are there. So because of the presence of air cavities, this tissue makes the leaf flexible. So mesophyll is made up of two types of tissues: palisade parenchyma, spongy parenchyma. And spongy parenchyma has got a lot of intercellular spaces, and these intercellular spaces makes the leaf flexible. And then third one, that is bundle sheath. You can see that the round structure, and inside the round structure you can see the bluish cells, and red cells, and these are the bundle sheath, or we say in simple way vascular bundle. And this vascular bundle consists of xylem and phloem. And as we discussed, the vascular bundle or bundle sheath is made up of parenchyma. But just above and below the vascular bundle, some parenchymatous cells or collenchymatous cells are also present up to epidermis. Okay, either parenchyma or collenchyma. And you can see here the presence of both xylem and phloem. And here the xylem is just present just by the side of the phloem, and you can see the numbers of vascular bundles also. And there is something called bulliform cells. There is no bulliform cells in dicot leaf, whereas in monocot we will see many. Let's move on to the next one. The epidermis, as we said, it covers both upper and lower surfaces. Upper epidermal layer is called adaxial, lower one is called abaxial. Cuticle is present. Upper epidermis consists of stomata and mesophyll. Upper and lower epidermis, it has got a lot of chloroplast, chlorophyll, photosynthesis I said, and it is of two types. Mesophyll is of two types, palisade parenchyma and spongy parenchyma. So palisade parenchyma is mainly pillar shaped cells. It's present top, adaxially placed, elongated cells, vertically arranged, parallel to each other. And mesophyll parenchyma, oval, and it is loosely arranged, as I said, amoeboid. Lot of air cavities are present, because of that flexibility will be there. And vascular system, it's seen in the veins and midribs. Size of the vascular bundle depends on the size of the vein. It's surrounded by thick walled bundle sheath cells, we said. And look at this, the anatomy of monocot leaf. It is basically isobilateral. Iso means similar, bilateral, both the sides, isobilateral. And stomata in monocot is present on both the sides, upper as well as lower, whereas in dicot we said stomata is present only in lower epidermis, whereas in monocot leaf both upper epidermis as well as lower epidermis stomata will be present. And what about mesophyll? Look at that, mesophyll tissue looks like one tissue, whereas in dicot we differentiated very clearly that mesophyll is made up of two tissues, palisade parenchyma and spongy parenchyma. Here you are seeing many amoeboid cells, so it's basically spongy parenchyma with large intercellular gaps will be there. And then in case of bundle sheath, it's made up of parenchyma, but just above and below vascular bundles are found sclerenchymatous cells. Now please look at this sclerenchymatous cells, whereas there we said it is collenchyma in dicot leaf. And let's see, there are three parts: epidermis, mesophyll, vascular. I want you to focus on this. Look at that. Now many mesophyll cells are there, amoeboid cells are there, and stomatal cavity is there, both the sides. Please observe the stoma, both upper epidermis as well as lower epidermis. And look at the xylem, primary xylem and metaxylem, and then xylem alternating with phloem. So this is how a monocot leaf will be present. So stomata present on both epidermises, upper as well as lower. Mesophyll is not differentiated into palisade and spongy parenchyma. And I said in dicot there are no special cells called bulliform cells which are responsible for throwing out excess water. Here bulliform cells will be there. They are modified adaxial epidermis cells and veins. It absorbs water and becomes turgid, and the leaf surface is exposed. It becomes flaccid due to water stress and leaves curl inward. So you can see here how the vascular bundle or bundle sheath will be present. So you got a xylem on top, and just below the xylem in the mid vein there is a phloem. So basically the bundle sheath is present in the veins, particularly mid vein. And how do you differentiate dicot leaf with monocot leaf? So nature and orientation. Basically we talk of nature and orientation. It's typically dicot leaf is dorsoventral, whereas monocot isobilateral. What about stomata? In dicot it is hypostomatic, usually on lower epidermis, whereas in monocot it is both amphi, both upper epidermis as well as lower epidermis. And similarly mesophyll, in case of dicot we are able to differentiate into two, palisade tissue and spongy tissue, whereas in monocot we cannot differentiate, and mesophyll looks everything alike. And the veins irregularly starred, and in monocot it is parallel venation. There we can say reticulate venation. And xylem vessels, many protoxylem and metaxylem vessels in each bundle in dicot, as you have seen in the picture, and 2 protoxylem and 2 metaxylem vessels in each bundle. 2 will be there. And bundle sheath is made up of collenchyma. In case of monocot it is made up of sclerenchyma. And as we said, the basic difference between dicot leaf and monocot leaf is based on orientation and venation. So as we discussed just now, it's basically dorsoventral in dicot, whereas in monocot it is isobilateral. And stomata, upper epidermis, monocot both upper as well as lower epidermis. Mesophyll differentiated into 2 tissues, palisade and spongy tissue, whereas in monocot everything is spongy parenchyma. Bundle sheath is made up of collenchyma. In monocot it's made up of sclerenchyma. And what about bulliform cells? Normally will ask you in your competitive exams. In dicot there are no bulliform cells, whereas in monocot they are present particularly in grasses. Hence, what is that you have to do? Now draw both the diagrams. Monocot leaf, TS of transverse section of monocot leaf, transverse section of dicot leaf, and tabulate the differences. And then just identify in your own garden how many dicot leaf you got and how many monocot leaves you have. With that, I am sure you will be happy to note the differences between dicot leaf and monocot leaf, at least morphologically. Once you come back to school, we'll see anatomy.